Welcome to the Novogene Solutions for Transcriptomics and Regulatory Events webinar. So I'm John Kafke, I'm Marketing Manager for Novogene Europe. So this is the first in a series of webinars we will be running. So we'll be publishing the full program shortly. So the webinar will last approximately 1 hour, and there'll be around and about 10 minutes of question and answer at the end of this. So please submit any questions via the Q&A text functionality, and we will endeavour to answer as many as we can. But without further ado, let me introduce your host, Caterina. So Dr. Caterina Baldi did her studies in medical molecular biotechnology in Italy and got a PhD in human pathology and molecular medicine at Sapienza University of Rome. She was a senior scientist in a genetic testing company for rare disease and was responsible for the somatic variant detection in cancer. Since July 2019, she is the Novogene Regional Account Manager for Brandenburg and Mecklenburg areas in Germany. Caterina, over to you.
Thank you, John, and good morning to everyone. This is Caterina Baldi, and today I will start giving you a general introduction of Novogene Europe. First of all, I would like to spend a moment to thank you for attending our webinar with such high participation, in this particular and difficult moment that we are all facing due to the pandemic. So let's start. Novogene was founded in 2011 and has been growing rapidly over the past few years. As you can see here in the picture, our work is focused to improve life science research, clinical diagnostic, and human health care, encompassing all applications of biology and life science. We have a strong scientific expertise and experience with 29 NGS-related patents, as well as over 4,000 research papers, including publication in the first-tier journals such as Cell, Nature, and Science. Novogene used the best and proven technologies from Illumina NovaSeq 6000, HiSeq, Oxford Nanopore, HiRent, and PacBio SQL platforms. Over the years, our state-of-the-art NGS labs have optimized various NGS protocols to maximize scientific outcome for your research. We also owned the largest sequencing capacity in the world with an annual throughput of 280,000 human whole genome sequencing samples. Our large assembly of NGS system enable us to run projects of any size, including those that requires massive amounts of sequencing and data analysis. Novogene has a strong global presence with established operation and laboratories in China, in the United States, in the UK, Singapore, Hong Kong, Japan, and the new one in Netherlands. We serve more than 30,000 customers in more than 60 countries globally and have extensive collaboration with many world-class universities, research institute, hospital, and biotech and pharmaceutical companies. Novogene delivers unsurpassed data quality to support our customer research goal. We offer a range of NGS-based service for animal and plant genomes, such as de novo genome sequencing or whole genome sequencing, transcriptome with mRNA sequencing, non-coding RNA sequencing, and single-cell RNA sequencing, microbial genome for de novo sequencing and resequencing, metagenomic sequencing, and amplicon sequencing. And for human genome, we offer whole genome sequencing, whole exome sequencing. And regarding epigenetic studies, we can offer whole genome bisulfite sequencing, chip sequencing, and RIP sequencing. Novogene Europe sales team is present in most of the European countries, as you can see in the map, and we are ready to support your project closely to you, discussing the meetings in person now on the website, and find the best solution for your research. At the end of this webinar, during the Q&A session, you will see a slide with all our faces and contact details. Our sales team is big. We are 21 regional account managers and very international with 15 languages spoken. And we can help you achieve the research goals. How? First of all, giving you the best informatic expertise and strict adherence to the quality control, and this results in the highest quality data. giving you also the publication-ready data if you decide to add the bioinformatic analysis to your project, selecting the most effective platform and strategies for your needs, and completing your project in a very short turnaround time due to our experience and professionalism and our NGS capacity. So now I would like to give a brief presentation of Dr. Bailey, who will present the webinar today. She did her studies at Clemson University in the US and got a PhD working on cotton transcriptome analysis. She started her career in Novogene as a senior bioinformatic engineer and then promoted to senior product manager for Novogene Europe. So thank you for your attention and enjoy the webinar.
Thank you for the introduction, Katerina. Now I'm going to share my screen. Can you all see my screen?
Not yet, Faye.
How about now?
Now, yes.
Okay. Hi, good morning, everyone. I'm Wei, and thank you again for your interest in Novogene service. And today I will give you a very brief introduction about the solutions we can provide for transcriptomics and regulatory events. In my presentation, there will be 3 parts. First, I will give you an overview about all the services we have for this topic, and then I will walk through the workflow of Novogene's service. And in the last part, I will give you some details about the specific services. Currently, more and more scientists are trying to answer questions using different omics strategies. If you are interested in the methylation level of the genome or the interactions between different macromolecules, you may want to perform the epigenome sequencing. If you are interested in the different types of RNAs, probably you want to perform the transcriptome sequencing. In some cases, when we talk about the transcriptome sequencing, we may refer to the messenger RNAs or coding RNAs. While in the cell, there are many other types of RNAs, such as the non-coding RNAs, including long non-coding RNA, small RNA, or circular RNAs. We also have the specific service to target each one. To take care of your project, I will go through the workflow for you here. First, we start with the sample preparation. For transcriptome study, that will be the total RNA. Here, I listed some recommended extraction methods. If you prefer to do that by yourself, you can use those methods as you need, or you can contact us for some extraction service details. So you may leave your message to us and we will come back with all the details. And next step, we'll move on to the sample quality check. Once we have our RNA ready, we'll go through those steps to check the sample quality. We will use gel to check if there is any potential contamination or degradation. We will also use nanodrop, Angelant, or similar instrument to check their purity and their integrity. On the right, this gel picture is showing some samples with protein or DNA contaminations, but we also have solutions for that, and I will talk that in details later. So for the Angelant, we will use that to check the RNA integrity number, which is the ring number. Angelant will give out a ring number ranging from 1 to 10. The higher, the better, or the higher, the less degradation. So 10 is the number we all want to get. However, if you have little degraded RNA samples, such as the ring number equal to 6, we can still work on it and enrich the RNA you are interested in. If you have any contamination in your samples, we will offer the purification solution. If you have super low input, such as the number showing here, we'll offer the amplification method. So we'll use the smarter kit, which will be shown in the next slide for the amplification. Only when we failed in both purification and amplification, we'll ask you to resend the samples. For the smarter amplification, we use SmartSeq V4 ultra-low input RNA kit. This kit is designed for single-cell RNA or ultra-low amount RNA. Based on their manual, it can be used for the RNA amplification from 1 to 1,000 intact cells. From our lab experience, we found it works well for RNA at least 1 nanogram, or RNA extracted from at least 100 cells. This kit can be only used for the eukaryotic RNAs, and if you have any samples have gone through smarter amplification, those samples cannot be analyzed together with other samples, as they have gone through the different protocol for the library preparation. After we have our samples ready, we will move on to the next step, which is the library construction. To target different types of RNAs, we have different types of libraries. For the messenger RNA from eukaryotics, they have poly-As. You can simply enrich them by using the poly-A enrichment method. So this library can be directional or non-directional. For some samples, like the messenger RNA extracted from FFPE samples, those samples has been fixed and stored for decades. So it is quite possible the messenger RNAs cannot be effectively enriched by using poly-A enrichment method. So in that way, we will propose the ribosome RNA depletion or removal library method. This method is also applied for the metatranscriptome method as well. And for some other non-coding RNAs without poly-A tail, or for the prokaryotic RNAs, so those RNAs, we will use ribosome RNA removed and directional libraries, as the strand information or strand sources is super important for us to accurately quantify those RNAs. For blood samples, they may have a lot of globin messenger RNAs in their input. So we will use the globin removal as well, combined with ribosome removal and the directional library for the blood samples. While for the small RNA, as they have very small insert size and they have their special ends, we will simply use the small RNA library method for this type of RNA. So what's the difference between the regular RNA library or the strand-specific library? The majority of the steps are pretty the same, except one step, which is the second strand cDNA synthesis. So we start from the beginning, which will be the poly-A enrichment, and then reverse transcription. During the second strand cDNA synthesis, we replace the DDP with UDP. So in this way, our second strand cDNA is masked with UTP, which can be digested by user enzyme. So for the sequencing, it will be very clear for us to tell if read one from the first cDNA or from the second cDNA strand. So that will help us to keep the strand information during the sequencing. For the RNA projects, as our expression levels will be highly affected by the sample status or environments, we will strongly recommend you to include replicas here. For normal RNA-seq projects, 3 will be minimum number. For some human disease-related research, we will recommend 5. And also for some better publications in the top journals, more replicas may be preferred. In our analysis, we will include the correlation analysis to help you double check if your replicas perform well or not. As shown in this figure, for each pair of the sample, we will present a correlation number or correlation index here. The higher, the better, or if they have very high correlation within each pair, that means those samples are very similar with each other. So from this correlation result, you can tell we have very nice replicas within each group, either control group or treatment group. And we have a very nice treatment effect or experiment treatment setup between treatment versus control. So this one will be also included in your analysis result or in your analysis report. You can either choose us to help you finish the library construction, or you can construct the library by yourself. But if you provide us the pre-made libraries, please also provide all the detailed information of your library. We may need to double check if your library is compatible to our sequencers or not. Once we have all the libraries are ready, we'll move on to the sequencing part. All samples or libraries will be sent to our Novogene Cambridge lab located in Cambridge Science Park. Over there, we have tons of experiences for different projects, and we will have unsurpassed data quality to ensure we deliver good data. And we also provide different pipelines or diverse pipelines to meet different requirements, either for the standard analysis or some customized analysis requests. After the analysis, we will give back the data to you. So we will deliver all the raw data. And for the projects without any bioinformatic analysis, we will also offer a data QC report. For the projects with bioinformatic analysis, we will also provide the analysis report and analysis results. So in the result folder, you will find all the results saved in Excel or in the PDF for the figures. So all of those figures are publication ready, so you can use them directly. And all the data will be delivered either in the Amazon Web Service or in the hard drive. So now let's move on to the next part, which will be some details. The RNA-seq for the eukaryotics is the major service we're providing in Novogene. And in this service, you can address the gene expression quantification and annotation of your coding messenger RNAs. For the library, we constructed the 250 to 300 BP insert cDNA library using poly-A enrichment method. And in here, I listed the data recommendation for different species. And the bottom part will be the sample requirements. So at last, I will list all the contact information for our local representatives. So if you need any more details about the sample requirements, you can feel free to contact them. For the analysis part, basically for transcriptome study or RNA-seq projects, the pipeline is pretty the same. So we start with the raw data quality control, and we map the clean data back to our reference, which is the alignment. And with that, we can quantify our rates to perform the differential expression analysis and the enrichment analysis. If this is all you need, you can stop here, or you want to move forward to study the structural information, including the alternative splicing, mutation, or fusion genes for human and mice species. If you don't have a reference genome, that's totally fine for the messenger RNA-seq. After the raw data QC, we will first perform the de novo assembling to construct the reference transcriptome. Then we can map all the short rays back to the unit genes to perform the following steps. We will also provide the annotation results using different databases to annotate your reference transcriptome. So good news, from our UK lab, now we are providing express messenger RNA-seq. You can receive your data within 15 working days. That will include library construction, sequencing, analysis, and data delivery. And this service will be performed on NovaSeq 6000 using pair end 150, maximally 24 samples. So if you are interested in this offer, also contact our local representatives. So everything I've talked in previous slides will be short read sequencing, like we use Illumina platforms. But right now, more and more scientists are interested in the long reads, which covers the full length of our transcripts. So PacBio is a very nice choice to perform the long-read sequencing. Compared to the short-read platforms, there will be no amplification or PCRs during the sequencing. That offers the PacBio platform with unique advantages, including long reads, high consensus accuracy, and high uniformity. So this will be unsurpassed advantages offered by the PacBio platforms. Right now, Novogene is the largest user of PacBio in the world. And every day we can produce more than 200 gigabytes data. And our average read length is higher than 14 kilobases per cell. So just feel free to contact us for more information. And using the PacBio platforms, we can offer the RNA-seq service, which is also the full-length RNA-seq. This is ideal for improving annotation in reference genomes, such as you want to characterize which is the exact isoform has been transcribed from my gene. So that means they can help you characterize the gene isoforms, alternative splicing, and fusion genes. Also, if you don't have a reference genome, RNA-seq can also avoid or overcome the short reads, the disadvantages from the short read sequencing or the incomplete transcripts. So for this, we will construct the PacBio RNA library, and we will use the fragment size selection method. Here is the data recommendation, and below is the data is the sample requirements. As I mentioned, there will be no amplification during sequencing, so that's why we ask for more RNA input. And to get or to cover the full length of RNAs, we will request a very high read number, the RNA integrity number. You can either only use the long reads to perform any structure-related analysis, such as give you the information for alternative splicing or for the gene isoforms, or you can combine those two services, using long reads for the annotation, for better annotation, and use the short reads from the Illumina platform for quantification analysis. So this will be a very nice combination with both annotation and quantification information. And in previous services I've mentioned, somehow they are sort of box sequencing, as they are mixing all the tons of cells for each library. But in this one, the single cell sequencing provided by 10x Genomics. So this is the real high throughput single cell RNA sequencing. As you can see in the figure, we separate the cell suspensions into individuals using the platform. And for each cell labeled in different colors, we attach the enzymes and buffers and anything needed for the future library construction. So in this library, it will give you two important information. It includes the barcode to help you track their cell sources, and it also gives you the UMI, unique molecular identifier, to track which gene this transcript is from. So with those two information, we can locate each transcript back to each gene or from each cell. So this is a very nice if you are working on cells and you really want to capture the heterogeneity of the cells. And the cell activity is super important for the expression levels. So that's why we cannot offer the library preparation right now. However, if you give us the pre-made library, we will follow up with the pre-made library sequencing and the potential analysis for you. So that will be everything for the coding RNA or messenger RNA. There will be other types of RNAs, as I mentioned, or showing in this figure. So those non-coding RNAs are highly involved in the regulatory of the messenger RNAs. And to target each type of those non-coding RNAs, we have specific services listed here. However, if you are interested in the network of multiple types of RNAs, we can also offer the whole transcriptome sequencing. We'll move on to each one to check some details. The first one will be the link RNA sequencing. In this one, it can cover or capture the information for both messenger RNA and long non-coding RNAs. Long non-coding RNA will refer to the non-coding RNA longer than 200 nucleotides. And to perform the analysis, we need the reference genome and the annotation available. For the library construction, we'll have the strand-specific and ribosome RNA removed library. And here will be the data recommendation, and below is the sample requirements. To perform the analysis, as I mentioned, for most of the RNA-seq analysis, the pipeline is pretty the same. We start with the raw data quality control. We map the reads back to reference genome. We quantify them and then perform the differential expression analysis and the enrichment analysis. As for most of the non-coding RNAs, they may be involved in the regulation of the gene expression. With that, we add in an additional step, which is the target prediction. For a link RNA, that will be the long non-coding RNA target messenger RNA prediction. So this analysis will allow us to look into the regulatory events between link RNA and messenger RNAs. Again, in the link RNA service, the standard messenger RNA analysis is also included. Next one will be the microRNA. As they have special ends and their small size, we will just provide the 18 to 40 base pair insert small RNA library. To perform the analysis, we also need the reference genome and annotation available. And again, they are quite small, so you no longer need the pair end 150 sequencing for that. For this, we will only use single end 50 for the small RNA sequencing. And here is the data recommendation. Below is the sample requirements. For the analysis, same pipeline, raw data QC, alignment, quantification, and differential expression and enrichment analysis. We also include the additional step, which is the target prediction. We use different databases for animals and plants. And in this one, we focus on the target genes of differentially expressed microRNAs and to look into their regulatory events. If any of those surveys cannot meet your requirements, or you really want to look into the networks between multiple types of them, so the whole transcriptome RNA-seq may be a better choice. You only need to send us the total RNA, and we will construct 2 different libraries using the same input. One is the ribosome RNA removal directional RNA library. This one will be used for the link RNA, messenger RNA, and circular RNA sequencing. Another library will be the small RNA library. Obviously, so this will be used for the small RNA sequencing. With all the data ready, we will include a joint analysis, which will include both correlation analysis between any 2 types of those RNAs, or co-expression network analysis among 3 types of those RNAs. So this is kind of a network study for the different types of RNAs. But one more thing I want to mention is this circular RNA library is the ribosome RNA removal and directional RNA library. It is different from the linearized RNA removal library. We may lose a little bit information for the circular RNA in this study. However, we want to focus on the network study or analysis in the whole transcriptome RNA-seq. If you really want to look into the circular RNA only, you can also send us the request for the linearized RNA removal library construction method. So here, I will go through the RNA-seq for the prokaryotics. So everything I've mentioned will be for the eukaryotics. Now is for the prokaryotics. And this service will focus on the transcriptome for the prokaryotics at a specific time or environmental condition. And again, we need a reference genome available for the analysis. This library will be also the strand-specific and ribosome RNA removal library, data recommendation, and sample requirements. As you can see from the right figure, the genome structure in the prokaryotics is different from the eukaryotics. So except the basic analysis steps, such as the data quality control or differential expression analysis, we also include the genome structure analysis, such as the new transcripts prediction, UTR analysis, or small RNA analysis. So this is specific for the prokaryotic RNA-seq. And that will be everything I want to share for the transcriptome part. And from here, we will move on to the epigenome part. Again, in this one, we really want to look into the methylation level of my genome, or I want to study the interaction events between my macromolecules. So that will be the epigenome sequencing. For the methylation study, whole genome bisulfite sequencing is the gold standard for the methylation sequencing. It can help us identify the methylation at a single base resolution. For the library, we will construct the bisulfite sequencing library, and we use pair and 150. For the data recommendation, we use 30x, which is the sequencing depth, as we are now talking about the genome sequencing instead of RNA sequencing. So that will be 30x of your genome size. And below is the sample requirement. This one is our analysis will be only applied for eukaryotics with reference genome and complete annotation available for the analysis. However, if you check the sample request, it is super high, especially for some applications or some samples extracted from cell-free DNAs or FFPE DNAs. So those DNAs will be super low input, which cannot meet our request. So that means we need to find a method for the low input DNAs. As I'm showing on the left, it is the library construction method for the traditional WGBS method or sequencing method. We fragment the double-strand DNAs, we add in the ligation, we add in the adapters, and then we perform the bisulfite conversion, which introduce a lot of DNA damage. That means not most, but majority of our DNAs will be breaking to pieces and which cannot be used for the future sequencing. While in the new method or in the low input method, PBAT, post-bisulfite adapter tagging method, we switch some of the steps. We perform the DNA fragmentation first, and then followed up with the bisulfite conversion. And then the third step will be the adapter ligation. So with this switch, actually we can use as much input DNA as possible because we can add in the adapter for majority of the converted input DNAs. So with this method, it can significantly lower down our sample requirements. However, this method or any samples gone through the PBAT method cannot be analyzed together with the regular samples. Here I listed the sample requirements from traditional WGBS showing on the bottom and the sample requirements from the PBAT showing on the top. You can see the sample request jumped from 5 microgram down to 20 nanogram for some of the samples. So this actually lowered down a lot or significantly for some of the very precious DNA inputs. For the analysis, very similar, that we perform the raw data QC first, and we map them back to our reference genome. As this is for the methylation study, we will call out the methylation by using the genome information. And then we check the methylation level at different scales, such as the genome scale, chromosome scales. And we perform the differential methylation analysis for methylated regions and methylated promoters. And for the IP sequencing, or for the interaction study, we'll have two options. First will be the chip sequencing, and this will be used for the interaction study between DNA and protein. And the second one will be the RIP sequencing. In this one, we'll look into the interaction between RNA and proteins. In chip sequencing, it can help us to look into the transcription factor targeting region and the histone modification. While in the RIP sequencing, it will release the information for RNA stability and structure and the microRNA targeting region. Here I listed the sample, the data recommendations for the chip study, and here will be the sample request. Even though you may think the input requests are super high, but we have tons of experiences with the low input DNAs for the RNA, we have the low input samples for chip sequencing and RIP sequencing. So if your sample input cannot meet our request, and please feel free to contact us to see if there is any possibility for us to move on. To accurately call out the peaks or the region you are interested in, we will strongly recommend you to send the IP and the input as a pair. I will go through the details in the next slide for the IP and the input. So in this figure, I showed a very general pipeline for the chip sequencing. And in chip sequencing, we want to study the interested region of the protein, which can bind to the different regions in the genome. So once we have those complex, we'll fragment them into pieces. Some of the regions may have the protein of interest, but some of the DNAs don't have the protein banded. And then we use the antibody to immunoprecipitate those complex. With the DNA released, we will move on with the library construction and analysis. So for the analysis, we need to compare those released or enriched DNAs or IP versus our background, which is not non-enriched or the input DNAs. So by comparing our IPs to the inputs, we can check out or locate where the peaks are. That will be the peak calling in the analysis pipeline. With those peaks identified, we can identify or we can look into the motif, which is the conservative sequence within those multiple regions on the genome that will release the conservative information across multiple regions on the genome. So that's why after the peak calling, we will move forward with the motif analysis and the peak annotation or the following steps. So by now, those will be everything I want to share for the transcriptome and epigenome. As you can see on the right, we have very nice publication every year from Novogene. And if you are interested in any new publications, please feel free to go to our website and for the new informations. And with that, I will thank you everyone again for your interest in Novogene service. And then we will move forward to our next slide. This is our amazing sales team in Europe, and we will take the question and the answering part.
Thank you, Bailey, for your presentation. This is Katerina again. Now let's start the Q&A session. So as some of you have already seen, some of the questions has been already answered by our expert technical support. For the others, the experience of way is needed. So we will ask to air. Unfortunately, we have only 10 minutes for this session. So if some of the question will not be answered, we will reply to you as soon as possible. So let's start with the first one that is coming from Carlos Perez Sanchez, that he asking, can you analyze microRNAs from biofluid samples like serum and plasma with small RNA sequencing? And what amount of serum plasma would you need? Do you run the extraction? And how many microRNAs can you detect in these biofluids?
So for this, it kind of depends on the project's status or the sample status. For the extraction, we probably need to contact our TS team to confirm if we can carry the extraction service for those special samples. And if they have for the analysis part, it actually used for the eukaryotic microRNAs. And we can double check that for you with our BI team to see if we have the special pipeline for the plasmids or from the microflows.
Great,
I think
you.
I need to come back with details later.
Thank you. And then we have Joanna Bianchi that is asking, how would we send samples to analyze using 10x?
For 10x RNA-seq, you can send us the pre-made libraries. So those can be shipped in GIS to our UK lab. And once we have the sequencing data, and then we will perform with the analysis. Or if you already have the data available, we can also offer the analysis part.
Great. The next one is from Giulia Guiducci, that she is asking, could I have a comment about what are the advantage of choosing long-encoding RNA-seq plus messenger RNA-seq to investigate links between long-encoding and messenger rather than performing a whole transcriptome RNA sequencing?
So actually for the whole, you mean the whole transcriptome RNA sequencing, right?
Mm-hmm. Yeah.
Yeah, for the whole transcriptome RNA sequencing, all the individual analysis package has been included already. I'm sorry that I forgot to mention that. That means for the whole transcriptome RNA sequencing, we will return the individual package for link RNA sequencing and for the link RNA analysis and the circular RNA analysis, small RNA analysis, and another package for their network studies, which I included in the small table. But for the link RNA service, it will only cover the link RNA and the messenger RNA. So if you are still interested in other types of RNAs, such as the circular or small, then you'd better choose the whole transcriptome RNA sequencing. If you only care about the link RNA service will be enough for you.
Great, thank you. The next one is from Ellen Verbrugge, and she's asking, if you have genomic DNA contamination in RNA samples for RNA sequencing, what is the best option? Treat the RNA ourselves or send it to Novogene?
Normally, we recommend you to send us the purified RNA samples without any contamination, but we can also offer the RNA purification if you need. So you just send us the sample status or information to us, so maybe we can evaluate based on your sample information.
Great, thank you. The next one is from Rafael Fernandez Munoz, and he asking, after differential expression analysis, do you by default provide moreover raw data and the preset comparison figures, also counts data, normalized, non-normalized, or both?
So for the differential expression, we will, I would say for the RNA quantification, we will offer those types of results. One will be the normalized one. Usually we will use FPKM, fragments per kilobase per milli. So that is the normalized quantification table. And we also offer the raw counts, which is used for the differential expression analysis.
Thank you, Wei. The next one is from Shannon Nelson, who is asking, do you perform the chip or should we send the extracted DNA for that?
Yeah, I forgot to mention that as well, and thank you for the question. So usually, as for each type of the protein of interest, you have to order the protein and construct to perform the enrichment of the DNA. So we don't offer the enrichment. So what we can only accept will be the enriched DNA. So you can perform the chip, the immunoprecipitation in your lab, and then send us the released or enriched DNA. We will follow up with the library construction and the analysis.
Great. Flavia Genoa is asking, when you talk about 3 or 5 replicates, do you mean during the sample preparation or the RNA extraction or later?
We really recommend from the very beginning point, which is the sample extraction.
Okay.
So it also depends on the project objectives. So if for the, such as for the de novo RNA-seq, if you really want to assemble your transcriptome for a plant, we may recommend you to pick up the samples from different parts of the plants, such as the leaf tissue, the root tissue, and the stem tissue. And those will be the sample extracted from different parts. But if you want to include multiple samples as your treatment replicates, you may want to include different samples as well.
Thank you. Eva Grochowska, I hope that I'm pronouncing good your names. Sorry if I did a mistake. Is asking, what is the minimum sequencing depth for detecting long non-coding RNA?
For the RNA sequencing, we usually don't recommend based on the sequencing depth. So for the long non-coding RNAs, I think we will recommend from 9 gigabase to 12 or even 15 gigabase. As long non-coding RNA, some of the long non-coding RNAs with very low abundance in their samples. So the higher, the more sequencing data may give us more chance to discover those long non-coding RNAs with very low abundance.
Okay, thank you. Now we have Silvia Manrique Urpi. She has 2 questions. So the first one is, what is the sensitivity of RNASeq for RNA in terms of percentage of a particular splicing isoform regarding the whole population of messenger RNA in the same locus?
So this is a pretty difficult question for me to answer.
It's very detailed, yeah.
Yeah, very detailed project. So probably we can discuss this case by case. And it really depends on different factors like your sample status, like your RNA quality. If you got very bad RNA for the isoform, you may get very you may get data with not very high quality, and it is quite hard for us to compare how sensitive those 2 systems are. So we can discuss this in details, case by case, and we will write down the contact information of you and get back to you after the
Yeah,
webinar.
if I can add something, I think that this doctor, maybe it's better to pick up your face and pick up your regional account manager, and he or she will be very pleased to have a meeting with you, even on Skype or in Zoom, and discuss about details for you and try to find the best solution for your project, I would say.
Yeah, thank you.
And then we have Christian Ramos that is asking, talking about replicates again, please define them. Different extraction from the same cell source using distinct culture dishes or different animals or human samples. What do you mean about replicates?
So you are trying to extract RNAs from different cultures, different animal tissues, and you want us to recommend the replicates for your projects. So if I understand this question right, so as I mentioned, those will be kind of the biological replicates that we're picking up different spots from the same tissues. If I understand right, such as for the cancer study. So for each cancer tissue piece, you may want to choose multiple pieces from the same tissue. As I mentioned, for the bulk RNA-seq, when we mixing tons of cells together, it's quite hard for us to capture the heterogeneity between or within each piece or between the cells. So that's another reason we want to recommend more replicates. So I will say maybe multiple pieces from the same tissue if you really want to look in the tissue scale. If you want to look into the individual scale, I will say multiple individuals as your replicates.
Mm-hmm. And also this is connected to Israel Ikoye question, so about the technical replicates, if we have any suggestions for it.
So for some of the projects, it's really hard to say whether it is a technical replicates or biological replicates, because for some of the disease-related research, it's very hard for us to have the technical replicates, as it's very precious input. So we will accept both in general, because we are talking in the whole transcriptome sequencing scale. So for each case, maybe it's better for us to talk the project in more details to see which is a better choice for you.
Sure. So feel free to contact your regional account manager to discuss about your project in details. The last question for now is from Dr. Aleman, who is asking for differential expression analysis. How many factors can be compared? If I have 4 different genotypes and 3 development stages, for example, do Novogene compare all possible interaction?
We will have the we'll have the form set up for you to fill in which comparison group you want to include in our analysis. And for the combined factors, I need to double check with the BI team if we can include the combined factor analysis for you.
Great, thank you. So we don't have now any more questions, and the rest we will be answered by email and contacting you privately. So our time is finished for today, and I would like to thank you, all of you, for your time, and we hope you find this webinar useful. We will email you a copy of the slides deck shortly, and we will also send you the program of our future webinars. In the meantime, please stay in touch. You can follow us on Twitter, on LinkedIn, or you can get in touch via our website, novogene.com. We look forward to see you all on the next webinar. And thank you all again and have a nice day.